Hi folks, in this video we will be talking about Taqman probe quantitative PCR that has three basic principles: denaturation, annealing, polymerization and stand displacement. This process begins inside the PCR and it begins with the denaturation step where the temperature becomes 95 degrees Celsius. First of all, let me just take out my laser pointer. So this is the temperature, and let's suppose this is a double helical DNA strand that joins both of the single stranded DNA molecules along with hydrogen bonds. And when inside the PCR, the temperature rises up and it becomes at least 95. So the hydrogen bonds between these two strands become broken, and here we can have a pair of single stranded DNA molecules. Afterwards, each of the single stranded DNA molecule is actually having a particular DNA sequence that is exactly complementary to the primer what we use to anneal the daughter strands. So this is the primer and it attaches along with the 5' to 3' end, right? Where its specific sequence is present. However, it happens at almost 5 degrees Celsius, but its temperature varies depending on the GC content and the length of the primer as well. On the other hand, here you can see a probe that is actually the Taqman probe. First, it is an oligonucleotide and it contains two main sequences at either of its end. First is the reporter that emits photons and another is the quencher that stop the reporter from emitting the photons under the normal condition when it is not bound at its particular specific position uh, with which it is complementary on the single stranded DNA, and it always binds after the primer position, right? It is understood that where the primer is binding right here, so probe will bind after that particular position. This happens because the companies from where the primers and these probes comes, where they are manufactured, they actually manufactured these primers and probes targetedly in a way by keeping the sequence that is to be amplified in mind, that the primer should come first. Afterwards, there has to be a place where the annealing will be proceeded in the form of polymerization of the daughter strand, and then they can have a sequence that is exactly complementary to the probe where the probe will be attached. And why they do that, we will see in the next slide. Okay, here we have the step of polymerization and stand displacement. So here you can see this is the primer that has already been attached in the step of the annealing in the previous slide. With the purple color, you can see this is the Taq polymerase enzyme that is proceeding for the extension of the annealed DNA strand along with the primer, right? So after the primer, we have the action of the Taq polymerases, right? And this is performing its action in the direction of 3' to 5'. Here you can see the parent strand belongs to 5' to 3' direction. However, the direction of synthesizing of the daughter strand is exactly opposite to it, right? So it would be 3' to 5', and it is working itself uh, in the direction of 3' to 5', just like that. And suddenly, in the passage, when it finds that there is another oligonucleotide sequence that is present in the 3' and 5' direction as well, so it will chop it. It will chop it because it is coming in its way, and it will chop it, due to which, let's see what happens. It will cut the reporter dye and the quencher sequence apart. Okay, so when it cleaves them apart, it is called exonuclease activity, right? That is also utilized for the proofreading purpose as well. So this is what happening, photon is emitting, right? And when the photon is emitted, light is detected through the detectors that are already present in the PCR. The detected light is converted or transduced in the form of graph plotting. So let's have a look, how does it look like. So this is a graph, right? Um, the red line shows the threshold, right? And after the threshold line, when we have a peak, so these peaks, all of them are showing the presence of that particular gene that we meant to amplify in the beginning, that we were actually amplifying. So if there would be a line, uh, let me just show you by using a pen. So if there is a line, right, if there is a line who is running just below the threshold, right, so this is considered as negative because it is not crossing the threshold. And uh, the reason why there is a peak that is below the threshold, it shows any sort of artifacts or any sort of chemicals that are present in your PCR tube, but it's not the DNA you want to amplify. This is not the targeted DNA. If it would be a targeted DNA, then it has to be, cross the threshold line. So this is how a plot of quantitative PCR looks like. And each different color shows the different gene, right? I hope this presentation helped you guys. Thanks for watching.